Thank you, Nikhil, and a very good evening. I can see that you just got up from your exhaustive. I guess your yesterday's day was quite exhaustive. That's why you're looking so tired. So, anyways, without wasting much time, we'd like to start my presentation. So, this is a session which has been sponsored by Pfizer, and I'll be talking on Lorlatinib as first-line treatment option in an ALK positive NSCLC. Yeah. So, as we all know, worldwide lung cancer is the second most commonly diagnosed cancer and the leading cause of cancer death in 2020. It represents almost 1 in 10 cancer diagnosed and 1 in 5 deaths as far as NSCLC is concerned. It almost accounts for 80 to 85% of all lung cancer cases, and the 5-year survival rate is really dismal. It is overall it is 25%, and for a distant metastasis, it's hardly 7%. As we all know that we are moving from conventional chemotherapy to targeted to immuno. Likewise, from pathological point of view, we are moving from initial histopath to molecular to now precision diagnosis. So treatment selection is also moving from being initially histology driven now to a targeted or oncogenic driver. So I think lung cancer is the most oncogenically addicted tumor, we can say, because whatever mutations we see in lung have not seen in any other type of cancers. So it is lung is an oncogenically addicted cancer, and we know that based on this, now our treatment is primarily tailored based on the oncogenic drivers, initial histology, and then oncogenic driver. So this is basically a slide which shows the lung cancer incidence in India. So you can see this estimated number of incident cases from 2018 to 2040 in lung cancer for both sexes and all ages. So you can see by 2040, almost the incidence of lung cancer is 1,19,370. And if we see the lung cancer mortality, then again by 2040, it's almost like 1,11,804 lung cancer mortality. So it is really, really very huge. And this is the data, so Globocon data 2018. So as I already said that, lung cancer is oncogenically addicted tumor. So as we all know, there are a lot many mutations. So ALK mutation is the second most common targetable genomic change mutation in lung cancer after EGFR mutation. And you can see this is a paper, original article published by Amanjit Bal et al. That is ALK gene rearranged lung adenocarcinomas, the molecular genetics and morphology in cohort of patients from North India. And you can see that after EGFR, the most common is ALK, which comprises around 7.9%. And then followed by the rarer ones like ERB-B2, BRAF, PIK3CA, NRAS, MET, MEK1, and no oncogen driver identified in almost 36% of the patient. So it is the second most common mutation. So, yeah. If we see ALK rearrangement in lung cancer, they are typically adenocarcinoma histology. They are typically, but not always, patients are never smokers or light former smokers. It tends to occur at younger patients, and there are diagnostic techniques now available for the identification of ALK mutation, and EML4 ALK and EGFR are mutually exclusive. So you can't find both the mutation in a single patient. So how there's a discovery of this EML4 ALK fusion in NSCLC? So what exactly is ALK positivity in NSCLC? So basically EML4 ALK fusion in NSCLC is nothing but a type of gene rearrangement, which we call it as an inversion, that occurs when a chromosome is broken in two places and the resulting piece of DNA is reversed and reinserted into the chromosome. So you can see this, there's a typical gene rearrangement that happens, that is called as inversion. And when this chromosome is break into two pieces, the resulting piece of DNA is reversed. So this piece is reversed and this get reinserted into the chromosome. And what we get is the ALK positivity. So EML4 ALK fusion was initially reported way back in 2007. And so if we see since we detected this EML4 ALK translocation in 2007, there starts the, ALK inhibitor development timeline from there. So 2007, we detected this translocation. In 2012, crizotinib, it is the first generation ALK inhibitor which has been approved in NSCLC. Then in 2015, the second generation ALK inhibitors like ceritinib, alectinib, and brigatinib has been approved Brigatinib 2015, 2017. Again, then you have second generation ALK inhibitors like alectinib and ceritinib was approved for first line use. Again, in third generation, then in 2019, Lorlatinib has been approved and has been discovered. And then in 2021, US FDA gave approval for Lorlatinib in frontline NSCLC with ALK positivity. So you can see 2007 detection, then 2012 first crizotinib, then ceritinib, alectinib, then second generation again, then 2019 third generation Lorlatinib, and 2021 we get Lorlatinib as FDA approval. So what are the current treatment options for ALK positive and why there's an unmet need for ALK inhibitor in these patients? So as I have initially mentioned that ALK positive NSCLC patients are generally young. The median age is around 50 years and they are non-smokers or light smokers. So CNS metastasis is very common. Almost 20 to 40% of untreated ALK positive NSCLC patients have CNS metastasis at diagnosis, and they are associated with poor prognosis. Also, it's not only just a CNS metastasis, but emergence of resistance mechanism that includes ALK resistance mutation and CNS metastasis. Due to this inadequate penetration of some currently available treatment options, continues to be a treatment challenge even with availability of second generation ALK TKIs. So there is a need for additional ALK TKI that in untreated patients prevent the emergence of resistant subclones and with improved CNS penetration. So we need good NSCLC ALK inhibitor for two reasons. One is for the CNS penetration, second thing to overcome the ALK resistance. So Lorlatinib, it is a third generation ALK TKI, which is a selective ATP competitive, highly potent third generation ALK TKI. It has excellent brain penetration to address the brain metastasis, almost 75%, and it has the potential to improve clinical outcomes with overall and intracranial activity in patients with previously treated ALK positive advanced NSCLC. In addition, upfront treatment Lorlatinib could completely suppress or at least significantly delay target resistance, leading to more durable clinical benefit, and that is what is the unmet need for ALK positive NSCLC. So what exactly is the Lorlatinib design and what is the precise mechanism of action? So it was basically designed to address this unmet need in ALK positive NSCLC, but metastatic brain disease and acquired resistance to the first or second generation ALK TKIs. So I know this is a very complicated biochemical structure of Lorlatinib. So you can see this is the crizotinib structure and this is the Lorlatinib structure. So this so basically this, if you see the crizotinib structure, and if you go to this, and this is the Lorlatinib structure, where there is, in addition to the this molecule, it has additional N minus derivative, which is there to overcome the resistance mechanism and for penetration. So with this effective CNS penetration and retention, and it almost binds with high affinity to most ALK resistant mutations. Now coming to the trial, that is CROWN trial, how Lorlatinib has been got the FDA approval in advanced ALK positive lung cancer. So this is the CROWN trial wherein they took patients who have stage 3B to 4 ALK positive NSCLC with no prior systemic therapy with a good performance status of 0 to 2, ECOG PS 0 to 2, who have asymptomatic treated or untreated CNS mets and more than one extracranial measurable target lesion as per REGIS criteria with no prior radiation required. They have been randomized in 1:1 with Lorlatinib 100 mg versus crizotinib 250 mg. They were also stratified based on presence of brain mets and ethnicity, Asian versus non-Asian. And the primary endpoint of this trial is basically progression-free survival by a blinded independent committee, whereas the secondary endpoints are PFS by investigator, overall response by again blinded independent committee central review, and intracranial overall response rate, duration of response, and intracranial duration of response, and OS safety and quality of life. So there's no crossover permitted between the treatment arms. So a total of 296 patients were randomized. It is a multicentric study wherein 23 countries and 104 study sites were included. And the patient was randomized from 2017 May to Feb 2019. A total of 149 to Lorlatinib and 147 to crizotinib. 5 patients in the crizotinib arm did not receive treatment, and all these patients underwent restaging CT scan and brain imaging every 8 weeks. And by 2020, almost 103 patients in the Lorlatinib group out of 149, 31 patients in the Crizotinib group remained on study treatment with a median duration of follow-up for the progression survival was 18.3 months and 14.8 months respectively in Lorlatinib and Crizotinib arm. So baseline characteristics are well balanced in both Lorlatinib and Crizotinib arm. You can easily see brain metastasis at baseline almost equal number, 26 versus 27, prior brain radiotherapy 6 versus 7, and stage 4 90 versus 95%. So coming to the primary endpoint, that is PFS by blinded independent committee review. So you can see there's a clear-cut 12-month PFS rate, Lorlatinib 78% as compared to 39% in the Crizotinib arm. So you can see median PFS is not estimable at 12 months, whereas it is 9.3 months in the Crizotinib arm with a hazard ratio of 0.28. Similarly, if we come to the PFS by investigator, which is a secondary endpoint, again, Lorlatinib median PFS is not estimable, whereas it is 9.1 months in the Crizotinib arm with hazard ratio of 0.21, again signifying a very good PFS benefit with Lorlatinib. So if we see the progression survival across the subgroup, there's a subgroup analysis irrespective of the presence or absence of brain mets, ethnicity, ECOG, age, smoking, and histology. It clearly favors Lorlatinib as compared to Crizotinib across the subgroup analysis. If we see the overall response rate, again it is better with Lorlatinib. 76% responders, out of which 3% patients have complete response, 73 have partial response, 13 patients have stable disease, whereas progressive disease is found in 7% of the patient. Median duration of response was not estimable, again, whereas it is in Crizotinib it is 11 months, and median time to response is around 1.8 months in both Lorlatinib as well as Crizotinib arm. If we see the intracranial overall response by blinded independent committee review, it is again those patients who have measurable or non-measurable brain mets at baseline. Again, the intracranial responders are better in Lorlatinib, 66% as compared to 20% Crizotinib, whereas patients who have measurable brain metastasis, again it is 82% versus 23%, with odds ratio of 8.41 in non-measurable, whereas it is 16.83 in the measurable metastasis at baseline. If we see the intracranial complete response rate, it is again better, 61% in the Lorlatinib arm as compared to 15% Crizotinib arm. Similarly, 71 with measurable brain metastasis and 8% only in the Crizotinib arm, again favoring Lorlatinib. If we see the intracranial time to progression, it is again median time to CNS progression is not estimable in the Lorlatinib arm, whereas it was 16.6 months in the Crizotinib arm with hazard ratio of 0.07 with significant p-value, again signifying the importance of Lorlatinib in this group of patients. So coming to the overall survival, definitely overall survival is not estimable in both arms, but you can see the hazard ratio is 0.72 in these patients. So coming to the safety summary, any type of grade adverse event is equal in both arms, whereas temporary disruption occurs almost similar, 49 versus 47%, whereas permanent treatment discontinuation is more with Crizotinib as compared to Lorlatinib, even though it is not statistically significant. Fatal adverse event occurs in almost similar patients. Serious adverse event occur in 34% patient as compared to 27, and grade 3, 72%. But fatal adverse event are almost similar. If we see the all casualty adverse event with more than 10% difference in frequency, again, Lorlatinib has high incidence of hypercholesterolemia and hypertriglyceridemia, whereas edema, weight gain, and peripheral neuropathy is more with again Lorlatinib, but it is not that significant. So if we see the global quality of life score, again it is better with Lorlatinib as compared to the Crizotinib arm. So to conclude, Lorlatinib resulted in significantly longer PFS, significantly high overall and intracranial response rate, and improved global quality of life compared to Crizotinib in first-line treatment-naive ALK positive mNSCLC. Lorlatinib intracranial response rate was 82% patient with measurable brain mets with a complete response rate of 71% and significantly longer time to intracranial progression. The safety profile of Lorlatinib was similar to that reported in previous studies, with grade 3 to 4 adverse event more frequent with Lorla than Crizotinib. However, the majority of laboratory abnormalities that were asymptomatic and readily managed. So these results really supported the use of lorlatinib as effective first-line therapy for patients with advanced ALK positive mNSCLC. And even the NCCN guideline did mention first-line options for ALK positive patients. Now you can see ALK reagent positive. If they discovered prior to first-line systemic therapy, then alectinib, brigatinib, or lorlatinib as category 1. Whereas if they are discovered during first-line systemic therapy, then either to complete planned systemic therapy, including maintenance or interrupt, and followed by preferred is lorlatinib, ceritinib, or brigatinib, or crizotinib. So coming to the hyperlipidemia and hypertriglyceridemia, which is more frequent with the use of lorlatinib. So it has been advised that patients should that hyperlipidemia is managed and resolvable through lipid lowering therapy and dose modifications, and one should not hesitate to use these lipid lowering agents if one is using lorlatinib on these patients. The CNS effect include change in cognitive function, mood, and speech. They are generally mild, intermittent, improved, or result upon dose modification. And these adverse drug reactions were reported frequency of almost 40%, with 12% of the patient experience more than one type of CNS effect. Baseline CNS matters was present in 72% of patients with CNS effects, and a total of 24 patients require more than one dose modification as a result of CNS effect. And out of these, 15 patients experienced a resolution of their CNS effect after temporary discontinuation or modification, whereas the median time to resolution of CNS effect was 12.5 days, and recurrence of CNS effects upon rechallenge at the same and reduced dose occurred in 6 and 7 patients respectively. So these are the dose modification guideline. So for a mild grade 1 adverse event, either you have to continue the same dose or withhold the dose until recovery, or rechallenge at the same dose or reduce dose by one dose level, that is by 25 mg. So of 100, one can use 75. Whereas for grade 2 to grade 3, withhold until the adverse event is less than grade 1 or rechallenge at one reduced dose level. For obviously grade 4 life-threatening, one has to permanently discontinue lorlatinib and instruct patient caregivers to report any changes in cognitive function, mood, or speech. Thank you very much. I'm sure I'm on time and would love to take any questions if at all there's. Thank you.
Thank you, sir. Yes, you were on time. So there's one question. Don't you feel there should be a study analysis of lorlatinib versus alectinib, as which one would be the best one to start one?
Well, see, if when I've gone through this lorlatinib data, it really looks impressive as far as CNS disease concerned. I'm not very much, comfortable with the resistant mechanism, but yes, CNS wise, if you see, 71%, rate, CNS control rate is too good if we compare. I mean, there has to be a head-to-head comparison between this alectinib and lorlatinib, and I'm sure one might do it, but for sure, as of now, we have just comparison with crizotinib. So the data looks really good as far as CNS disease is concerned.
Yes, sir. Thank you, sir. Thank you for your valuable time. And I hope it is kind of a practice changing here, where I think now we would start lorlatinib or alectinib as a first line. I next